I just wanted to give a quick overview of where we're coming from and where we're going. And I'm going to kind of fly through these slides to get done in about 10 minutes. But I think this is just kind of an overview that I think is helpful to get the perspective of why we're here and what we're doing. This is an old slide that shows the survival for glioblastoma, which is the primary disease we're talking about, with a biopsy alone, which is somewhere around 4 to 6 months, versus a complete resection or extensive resection, which is median survival, 6 months or so. When radiation came along, that doubled that survival to about a year. And then with chemotherapy, we got improvement in survival that was hailed as a breakthrough in 2005 by the introduction of temozolomide, which is really only beneficial to about a third of glioblastoma patients, shown in the just have a pointer on it. Shown in the slide to the right, the group that are methylated and the MGMT promoter, but that moves the curve for everyone. So after 2005, the median survival was about 15 months. Further investigations or studies have shown that, which have not received much publicity, that even more extensive resections aided by technologies such as neuronavigation and fluorescent imaging of tumors intraoperatively can help us extend our resections. And that actually is quite significant if you look at overall. So very aggressive resections can improve survival by a significant number of months. And then in the last few years, we've had the introduction of some unconventional therapies like tumor treating fields for the Novocure device, which has improved survival another 3 months or so on average. So now the approved therapies include radiation, temozolomide, and Novocure device for glioblastoma. But despite all these advances, the diagnosis is still associated with a dismal prognosis, with a median survival now in the range still of less than 2 years. So how do we move beyond these incremental changes, and how do we leverage our new understanding of the molecular biology of these tumors and new pathways? We have all of these tools at our disposal: surgery, radiation, chemotherapy, targeted pathway inhibitors, targeted molecular markers, and we have pushed the envelope on all of these as far as I can tell without making a major leap in advance. There are a lot of unknown aspects to what could be lying in the future with further research, and that's what we're going to talk about today. And hopefully some of these modalities will uncover, will turn over stones that show breakthroughs that will allow us to have a major improvement in survival. The last decade or so has been focused on pathway inhibitors. We can sequence the genes of these tumors and show that there may be pathways that are highly activated, and 20 years ago this was thought to be the Rosetta Stone for cancer. Just figure out which pathways are overexpressed and then knock them out with drugs. The drugs shown here in these little blue areas. Does this thing have a pointer on it? Am I missing something here? Oh, wait, here it is. Do you have a laser? Okay. Now you can clearly see. All of these drugs, exactly. All these drugs have been tried that can knock out these specific pathways, but we've come to realize that these tumors have multiple redundant pathways. You knock out one pathway, another pathway becomes the driver. There are certainly known to be molecules highly overexpressed on a percentage of these tumors, So you would say, well, why don't we just target those because their ratio of expression in the tumor to normal is very high. So let's make monoclonal antibodies that attack EGFR or the mutated version. And we've tried these in clinical trials, and they have not resulted in the significant breakthroughs that we would hope. If you treat a tumor with EGFR V3 antibody, the tumor may come back at some point without that protein expressed. We've had a big interest in discovery in glioblastoma stem cells, and this has led to some concepts that if these cells really are the drivers of the tumor, could you knock out their function by figuring out specific pathways like Notch signaling that will shove them from a tumor-promoting cell to a differentiating cell, which will result in decreased tumor growth. So a huge amount of interest in the last decade has been focused on understanding these elusive glioblastoma stem-like cells. And then I think in the last decade or even 5 years, an appreciation has really focused on the fact that these tumor cells are not in a vacuum. They are in an environment that is surrounded by this yellowish color here, in my mind, represents the force field of the immune system blocking the normal immune function. These tumor cells produce certain proteins like PD-L1 and other things. They produce cytokines like perhaps the cytomegalovirus cytokines and other ones like TGF-beta that essentially make the tumor an invisible environment. And they can reprogram T cells that are trying to kill the tumors to express certain molecules that will prevent the T cells from killing the tumors. That's led to a spy versus spy approach. If we can block these molecules with antibodies like CTLA-4 and PD-1 checkpoint inhibitors, we can maybe get these guys to turn back on and attack the tumor. So we have dozens of clinical trials now ongoing, some that we're doing here to try to treat patients with these checkpoint inhibitors. We've been investigating all types of ways to try to vaccinate patients against their own tumor. The DC VAX trial we've been involved with here and the ICT 107 trial are based on the fact that maybe if we can program the dendritic cells in a patient to activate against their own immune markers from their own tumor or important cancer stem cell markers, then we can drive an immune response specific to the tumor. So those are ongoing studies, and then a host of other types of approaches. As I mentioned, the spy versus spy approach to try to knock down the things that are knocking down the T cells have led to ongoing clinical trials with Opdivo and Keytruda from various companies that have been successful in other tumors like melanoma where there are really high levels of mutation. And these are ongoing studies with checkpoint inhibitors for glioblastoma in the setting of standard of care radiation and Timidar. Other immunotherapy approaches include adoptive cell therapies, adjuvant immunotherapies, monoclonal antibodies that target, for instance, the EGFR V3, and then insert into the cell, once they bind to the cell, a molecule that is toxic to the cell. So all kinds of strategies are being investigated, and I think some of the pot of gold at the end of the rainbow will probably involve finding specific things that are expressed on glioblastoma cells or stem cells, and then designing immunological molecules, or for instance, specific T cells that can be designed to attack those molecules so that the patient can have a specific immune response to the tumor without the side effects. And then there's a whole other kind of concept of infecting the tumor with a virus that proliferates within the tumor but not in other normal cells, and that virus may carry with it a prodrug or an enzyme that converts a non-toxic drug to a toxic drug, or the virus may kill the tumor itself, like the poliovirus approach at Duke, and then release a host of cytokines that will orient the immune system. I And as I said, we have currently standard of care includes now using Optune or the NovaCure device, which is an out-of-the-box concept which has alternating electric fields which sort of stuns the microtubules in the cells that are oriented in that direction and decreases proliferation of the tumor cells. We've been experimenting here with another device from a company locally in Seattle, which is using very low frequency radio frequency energy, very low energy radio frequency energy to mimic a chemotherapeutic drug, and the results here are quite interesting. But to move forward, I think we need to continue to have groups like this where people with all these different perspectives come together and sample what's going on in other fields, and hopefully by bridging different areas of investigation, make connections that can lead to putting these things together in a way that might have a synergistic advance. And that's what our group here is doing. We're very fortunate to be affiliated with, officially now, with the Institute for Systems Biology that Dr. Hood here has started years ago to try to use high throughput screening, for instance, of FDA-approved drugs to find out why someone's own tumor stem cells will be sensitive to that drug, and then use algorithms and genomics to figure out if you can sort of reverse engineer and predict why these drugs are working for certain patients' tumors. So we need to continue to think about the hallmarks of cancer and work together to see if we can bridge some of the information from immunological studies and genomic studies and high throughput screening to lead to more significant breakthroughs. And the whole field is moving toward a more personalized approach with targeted therapies. Maybe the high throughput screening will lead to unexpected discoveries, and maybe some of these other approaches, such as the radio frequency waves and alternating electrical fields, will feed in and maybe synergize with some drug therapies. So there's all kind of approaches that we need to consider. And before I introduce our speaker, I would just like to pay tribute to my former colleague who recruited me here, and for whom my directorship is named. So Greg Foltz was a leader in neurosurgery and neuro-oncology who started the Ivy Center here at Swedish. He had worked with Dr. Hood and Dr. Hood's group, and he was a visionary, and really even 10 years ago started an approach to personalized cancer therapies and high throughput drug screening, focusing on genomics and targeted immunotherapy approaches, which we're still pursuing today in the lab.